So here actually we're recording. So one more, welcome everyone to the Population Dynamics Virtual Seminar. So this Friday we have Seppe Kuehn from the University of Chicago. And the title of the talk is Simplicity from Complexity in Microbial Communities. A couple of words regarding the speaker of today. So he got a PhD in chemical physics from Cornell University, and he did postdoctoral research at the Rockefeller University in New York. Then he became assistant professor at the Center for the Physics of Living Cells at the University of Illinois. And now he's assistant professor in the Department of Ecology and Evolution at the University of Chicago. What are actually the questions and the topics in which Seppe is interested into? First of all, quantitative microbial ecology and evolution. So some questions that he would like to tackle are, for example, how do microbial communities function? What are the molecular mechanisms? And why has evolution resulted in the microbial community structure we observe in nature? And he investigates all these questions with different methods, both quantitative measurements, experiments, and theoretical methods. So with that, I would like to give the stage to Seppe, and thanks again the speaker to be here. And I'm really sorry for what happened last time because we were quite misunderstanding about the time. I will stop sharing my screen. No. I don't know if actually have you seen my screen.
I don't
it was
think
not.
No, we didn't see it.
It doesn't matter.
Well, it doesn't matter. The presentation was just vocal and oral. So, Seppe, please, you can actually share your screen.
Can you guys see my slides okay?
Yeah.
And can you hear me?
Properly?
Yeah, okay, let me just... Okay, great. Hey, it's so wonderful to be here. I'm sorry about last time in the time zone, but here we are, and I'm excited to tell you about some of the work that we've been doing in my group for the last 2 years. Please do interrupt. My training is in physics, and we interrupt in physics. So please do. I'm at the Center for the Physics of Evolving Systems at the University of Chicago, and my tenure home is the Department of Ecology and Evolution. So before I talk about microbial communities, I'd sort of like to take a step back and talk about biology at a much larger scale. And that scale is the biosphere as a whole. When we look at the biosphere as a whole, we notice a lot of certainly complexity that's apparent in this image. But at a certain level, the biosphere as a whole is actually very, very simple. When we zoom all the way out to the sort of biogeochemical level, one of the defining properties of the biosphere as a whole is these biogeochemical cycles. And those cycles are themselves really and truly remarkably simple. So here I'm showing you examples of the nitrogen cycle and the carbon cycle. And these biogeochemical cycles are driven by redox chemistry, which is mediated by, in large part, not entirely, but in very large part, by microbial metabolism. So these redox reactions, which transform, for example, carbon dioxide to sugar through photosynthesis, and the converse process converting sugar back to carbon dioxide through respiration, are at the sort of highest level of organization in the biosphere, quite simple objects. Similarly, in the nitrogen cycle, we have the transformation of nitrate to N2 by denitrification, which we'll talk about today, nitrogen fixation, and nitrification. And to the biochemists that figured out these metabolic processes that drive these biochemical cycles, the simplicity was quite apparent. In fact, the Nobel Prize-winning Hungarian biochemist Albert Szent-Györgyi declared in the process of discovering these metabolic reactions that life is really nothing but an electron looking for a place to rest. And the electron here refers to the fact that these reactions are redox reactions. So as the cycle proceeds, the redox state of either nitrogen or carbon is going up and down and up and down and up and down over and over again. Now, if life were just this simple, I think at least my department would probably not exist. And in fact, it's not quite this simple, right? And the reason that it's not this simple is that the ecosystems and the biospheres that give rise to these high-level biogeochemical cycles are immensely complex. So we know that the communities and the ecologies that give rise to these biochemical cycles have many, many components, that those components interact in a huge number of ways, and that there's just sort of an unbelievable level of diversity in these ecosystems and the biosphere as a whole. So in some sense, the way that I see the question in biology is what is the relationship and how do we understand the mapping between this complex level of organization and this very simple level of organization? I make this arrow here at the bottom two-headed intentionally because what I'm trying to ask is, how does this simple biogeochemical process come out of the insane complexity of these ecosystems? And similarly, how does such a simple process, such as this cyclic transformation of carbon, give rise to this seeming unending complexity that we observe in these communities? I guess I'd just like to note one contrast with physics. In physics, we are often interested in just one direction, really. How do simple rules, say Newton's laws, give rise to complexity? Or how do simple interactions, say in a quantum many-body system, give rise to very complex emergent behaviors? So in there, we're looking at how simplicity gives rise to complexity. And I think in biology, one of the fascinating things that we're doing that's very different is, how does complexity give rise to this simplicity? And so this is, I think, one of the reasons that I'm working on biology now. Okay, so at the level of the biosphere, this question is, of course, very important, and many people are working on it, but it's also very hard because making measurements, understanding the interactions in the system at this global scale is a huge challenge. At the same time, though, one of the exciting opportunities, I think, in the modern era is that microbial communities allow us to cut this problem down to size. And what I mean by that is that microbial communities exhibit a lot of this incredible complexity that we observe at this higher level of organization, but in a package that's microscopic, maybe tens to hundreds or thousands of microns, here in a soil grain, or here in a piece of organic matter in the ocean being degraded by microbes. Yet these microbial communities harbor this similar levels of complexity, I would argue, and I'll get more into what I mean by that in a moment. And at the same time, they drive these processes, these transformations, these redox-mediated transformations in a way that is not only similar to the global biogeochemical cycles, but in many cases responsible for those cycles. So in this example that I'm going to talk about more today, this transformation of nitrate to N2 by denitrification, essentially all of this half of the nitrogen cycle on the planet is done by bacteria. And so not only are these communities tractable systems where we can ask this question, but they're also relevant for this higher level function of the biosphere. Okay, good. So I'd like to say a little bit more about what I mean about this complexity. When we look inside, let me just adjust this. When we look inside one of these microbial communities, say here in a grain of soil, this complexity is apparent really at multiple levels of organization. So the sequencing revolution has shown us that the taxonomic diversity in these communities is really substantial. When we sequence a gram of soil, for example, what we find is 10 to the 4, 10 to the 5 taxa. And those taxa all possess genes. These are supposed to be little bacterial genomes. And those genes are organized in really complicated and diverse ways on these genomes. That's simply at the structural level, but those component parts of these microbial communities, these different taxa, they also interact with each other in complicated ways. And this is, of course, the purview of really the last century of community ecology. And finally, out of all of this complexity comes this sort of emergent functional process that we're interested in trying to understand. And so I would argue that zoomed in at this level, there's a similar relationship between the underlying complexity of the system and the sort of emergent simplicity of the system in terms of these functional processes that I argued for at the biosphere, at the level of the biosphere. And so what my research program is trying to do is to try to connect this underlying complexity to this simplicity, and as I said before, go sort of both ways. So as I've stated it now, I don't think there's really a sharp question here. I've said we want to relate something about complexity at this underlying level to these sort of simple phenomena at the functional level. But I think this is not a sufficiently sharp question for one to design a research program, or certainly to go into the lab and do some experiments. So how can we take this question and make it crisp enough such that we can go into the laboratory, into nature, and do experiments? And I think one tried and true way to take a question like this and turn it into an actionable question is to pose it as a design or prediction problem. So the fundamental question really is, can we design or predict community function? And let me just define very clearly my terms here. The function of the community is a shorthand word for, say, a metabolic transformation that the community is responsible for. It could be the degradation of a compound. It could be the suppression of a pathogen. It could be any number of processes. And I don't mean this teleologically. I don't mean this as if the community is intending to do this in a sort of evolutionary or fitness sense. I mean it as a shorthand. But of course, the community also has this high level of taxonomic and genomic diversity at this structural level. And this is transmuted through these complex nonlinear dynamics to this functional level. And so what I would like to pose in this talk, and really in most of the projects in my lab, is can we design and predict community function? And what are the necessary ingredients to do that? So I will first argue to you that this problem is not easy. And it's really not easy for three primary reasons. The first reason that I've already talked about is structural diversity. So I already mentioned that when we run the sequencer on soils or on human guts or in oceans, there's just an incredible amount of diversity in the system: large taxonomic diversity, allelic diversity, genomic structure diversity. And we don't really honestly know how much of that diversity matters for the dynamics and ultimately the functional property of the system. The second reason this problem is hard is heterogeneity. And in this system, heterogeneity manifests itself as environmental heterogeneity. We know that the microbial communities all over the world are present in a huge number of different environmental contexts. And what that means is that you may have a similar community at the structural level, but they're in a different temperature, pH, oxygen concentration, and so on. That's going to affect these interactions and then ultimately this functional property. So that's hard. And finally, these two, this diversity and heterogeneity is filtered through nonlinear interactions in the community that ultimately give rise to this functional property of interest. And so that makes this problem not easy. I think I learned as a PhD student that not all hard problems are worth solving. Sometimes we spend a lot of time and effort solving hard problems and they're not even that interesting. Trying not to do that. So I'm going to argue to you that this problem is, not only is it hard, it's actually something that we should do. And we need an answer. I think it's actually very easy to argue in this context that we need an answer. The reason is that these microbial communities are responsible for host health. They have huge impacts on global climate. So for example, methane production by cows. In soils and in oceans, the production of greenhouse gases such as nitrous oxide or carbon dioxide are super important at a global scale. Just to give you an idea of the carbon budget, 10 times, if you take the total carbon dioxide produced by all human activity, photosynthetic microbes in the ocean are fixing about a factor of 10 more per year. So this is not a small effect. These are very, very important processes. Each one of these problems are things that we're either working on in my lab or collaborating with other people to try to understand. So with Arjun Raman, who's a quantitative biologist and pathologist at UChicago, we're looking at designing communities for therapeutics and environmental remediation with my student Mahmoud Youssef and an incredibly talented postdoc, Rita Oliveira. Kyle Crocker in my group is doing theory to help Itzhak Mizrahi understand methane production in cattle. I'll touch on this briefly at the end, but my student Ki-Suk Lee from UChicago is working on understanding the fluxes of metabolites through very complex soil microbiomes using a combination of experiments and theory. And recently we've started a super exciting collaboration with Otto Cordero at MIT, a good friend, powered by a postdoc, Vaibhav Sinha in my group, trying to understand carbon remineralization in microbial communities. So this is just a glimpse of some of the projects that we're working on. I won't talk about them in detail today. So I've argued to you that the problem is hard, and I've argued to you that we need an answer, but that doesn't mean that we can solve it, right? So we need a strategy to address this question. And so the question I'd like to ask now is, what is the strategy that we're going to employ to solve this problem? The strategy is, again, a three-pronged strategy that we use in almost all of the projects that are going on in my group. The first thing to do is to use a statistical approach to try to cut through the diversity and the environmental heterogeneity in these systems, to let the data tell us what matters in terms of which environmental variables are affecting community dynamics and function, and to let these statistical approaches, which I'll articulate, I think, very clearly in this talk, tell us what aspects of the diversity in the system actually matter for functional properties of the system. The second ingredient that we bring to bear is simple models. And I mean the word simple very, very literally. None of the models that we're using are complicated. They're all sort of pedestrian at the level of sort of an undergraduate in theoretical ecology or physics or applied math. So these simple models act as null hypotheses for understanding which aspects of these nonlinearities are actually important or not for the function. And so there's no fancy math, but the math is an important tool for determining what matters. Neither of these things would work if we didn't have quantitative measurements of the function. And so we spend kind of a horrifying amount of time getting these measurements right, because of course, if you're not measuring the functional property of the community of interest correctly, then these things won't help you at all. So this is the strategy for solving what I think is a very hard and important question. Okay, so the goal for today is to take you through three short stories where I take this strategy of employing these three processes to solving this question of relating the structure, the environmental heterogeneity, the dynamics in these systems to function. And so I'll start with a very brief summary of two papers where we connect the taxonomic structure of a community, that is who's in the community, to the function. And then I will walk you through a paper published last year, which maps structure to function in a particular context. And finally, I'll present recent work that we've just submitted, which lets us look at how environmental heterogeneity in wild communities affects the structure of those communities. And finally, I'll just show you one example of what we're trying to do in the future, just very briefly, one slide. Okay, I was assuming there's no questions. I can't see the chat or anything, so I will just keep going. Okay, so here's the first question. How does taxonomic composition determine function in microbial communities? So imagine I have a microbial community. Here's my hand-drawn cartoons. They're interacting in these complex ways. And they're performing some transformation of compounds, say nitrate to N2. And now I go in and I delete the orange species. This presumably changes the community in complex ways, and that somehow changes the function of the community. And what I want to know is, how do I predict what's going to happen here? How do I understand this? This looks hard. I think the sort of standard way of doing this is to try to dissect all of these arrows and these interactions, which is, of course, not simple in a complex community. And so what we wanted to do was to try to approach this question in a slightly different way. And the ideas for approaching this question in a slightly different way, which involve not understanding all of the mechanisms and interactions in the community, but instead treating this community as if it were an organism in abstract, not in reality, and thinking about it in the context of a fitness landscape. So this is an idea that comes from old papers in my group, from conversations with Alvaro Sanchez, Mikhail Tikhonov, Arjun Raman, many people, where we wrote a perspective last year, and a recent paper was just accepted this morning, actually. So the way we're thinking about this is as follows. Think about a fitness landscape where you have some fitness, and in this lower plane, we typically think of genotypes, the presence and absence of different alleles in an organism. So what we're doing in this sort of conceptual reframing of this structure function problem in communities is to imagine this z-axis as a function of a community, and points on this plane as the presence and absence of different taxa in the community. So as I move around in this plane, I'm moving around in a binary sort of genotype of the community, which is telling me which strains are present and which strains are absent. And so what we did was we took data where synthetic communities were available, where people had constructed different synthetic communities where the composition, the presence and absence of that community is known, and some functional property is measured. A beautiful data set from Ophelia Venturelli on butyrate production, and some other data from my group and from Alvaro's group. And what we did with these synthetic community data sets where we know which strains are present and absent was we simply fit a linear regression, which is familiar to everyone who does quantitative genetics, where the function of the community is a response variable, and the presence and absence of each strain in the community is indicated as the independent variables. So sigma I is 0 or 1, depending on whether or not the strain is present or absent. And then we have epistatic terms, which indicate a statistical interaction between the presence and absence of two strains. What we show in this paper is that this incredibly naive model, which really considers no dynamics, no interactions, just considers the presence and absence of every strain and any epistatic interactions between those strains, gives you kind of incredible predictive power for the function. So here I'm plotting the predicted butyrate production for Ophelia's data against the observed butyrate production with an R-squared of almost 0.9 using only second order terms. What we show in this paper is that the reason, at least, that this works in these examples is because the landscape that we're inferring here is not rugged. And what I mean by not rugged in this case, which rugged is a term that's hard to define, has many definitions. What I mean by not rugged is that these additive terms dominate this landscape. So most of the variation in the structure of this landscape is coming from simply the presence and absence of individual strains. There's more work to do here. We have more work going on in this space, but I think this is a new way of thinking about this problem. Part of this thinking comes from work that we did several years ago on closed microbial communities. These are self-sustaining microbial ecosystems where we hermetically seal them in vials, and we developed a sort of beautiful quantitative measurement for quantifying the rate of carbon cycling around a carbon cycle in the community. So this community has some bacteria that do respiration. It has an alga that does photosynthesis. And when you seal them up together, they sustain a carbon cycle for months and even years. And what we can do is we can measure the rate of that carbon cycling. And in this PNAS paper in 2021, what we showed was that as you vary the composition of the bacterial community, so we're putting in random different compositions of the bacterial community that are responsible for this side of the carbon cycle, that variation in that composition doesn't affect the overall rate of carbon cycling in the community strongly. So there's sort of a degeneracy, a redundancy in the system. And so what that means is that going back to this functional landscape where now this z-axis is the carbon cycling rate of the community, what that means is that many different arrangements of this bacterial component of the community give rise to the same function. And so this mapping from taxonomic structure in the community to function has a very high level of degeneracy in the system. So there's much more work that we're doing here on understanding these functional landscapes of these communities and asking whether we can use these approaches to really design communities to get jobs done. And that's something that we'll be writing more about in the next year or two. Okay. So I'd like to transition now to part 2 of the talk, which will be a more detailed investigation of a paper that we published last year. And that paper addresses the following problem. The problem is, I just showed you how taking out a strain affects the function, how we can make this prediction. But what I just showed you knows nothing of the genomic architecture of those strains. So we know these strains have many different genotypes. And how can we connect this gene level variation in the community to the function? And in the spirit of the framing of this talk, I'm going to argue that we need to pose this again as a prediction problem where we go from genotypes to phenotypes or traits of members of the community to interactions and ultimately to function. And so the question is, the way this project really started many years ago now, unfortunately, is how much information about the flux of metabolites through the community is encoded in the genes that are present in that community. You know, one could imagine that there's not much information, that you can't make this prediction. And that was certainly a possible outcome of this project. It's not what I'm going to show you today. What I'm going to show you today is that there is a large amount of information encoded in just the genes that are present in a community about this flux of metabolites. And I'm going to show you this in the context of a particular metabolic process called denitrification, using, again, a statistical approach and simple models, like I talked about before. Okay, so I will use denitrification as a model metabolic process. This has been a powerful model system for us to ask basic questions in microbial ecology because the metabolism is very well understood and because many of the molecular details have already been worked out. So denitrification is anaerobic respiration. So what that means is that if I have organic carbon available and I'm, say, Pseudomonas aeruginosa, and oxygen is not available, the environment is anaerobic, I can use nitrate, nitrite, nitric oxide, nitrous oxide as electron acceptors. So I'm taking NADH from TCA cycle and glycolysis and so on. I'm donating electrons to these oxidized nitrogen compounds, reducing them, and running my electron transport chain with this process. This drives a cascade of reactions from nitrate all the way to N2, which is very important in the nitrogen cycle. And what's remarkable about denitrification is that it can be a collective process. So this pathway can be split up across strains in a soil environment, for example. So you can go isolate organisms that do just the first step, organisms that do the last 3 steps. You can actually find organisms that do step 1 and step 3, but not step 2. All sorts of combinations are available. And some organisms actually do all 4 steps. Okay. So the point is that the details of this are understood. The other salient feature is that these organisms are easy to isolate and culture in the lab, which was a requirement. And we can easily quantify the metabolite dynamics for at least these first 2 steps. So these 2, nitrate and nitrite, are gases, or are soluble, excuse me. Nitric oxide and everything downstream is in the gas phase. Okay, so what Karna, my postdoc Karna Gowda, who will be starting his lab soon, I won't announce where yet because I'm not sure he's done it, but Karna, who you should all go do postdocs with, an incredible scientist and human being, what he did was he went to the cornfields and the soybean fields outside of Urbana-Champaign, Illinois, dug up some dirt, plated it, and then screened these strains for denitrification. It turns out something like 5 or 10% of cells in a soil sample do this process in some capacity. And so here's a phylogenetic tree of the 80 or so isolates that he isolated. Those isolates have 3 different phenotypes. So one phenotype, which I'll call NAR-NER and denote in purple, performs both of the first 2 steps in this denitrification cascade. So here I'm plotting nitrate and nitrite, nitrate in blue, nitrite in red, and I'm plotting the metabolite dynamics, not the population dynamics. And what you can see is that nitrate is consumed in this blue curve, nitrite is produced and then consumed, okay? The second phenotype is a NAR strain, a nitrate reducer that does just the first step. Here you can see it reducing nitrate, producing nitrite. And the final phenotype just does this second step of the reaction as shown in this plot. So what Karna was able to do is in individual cultures of 80 different isolates was to quantitatively phenotype them by measuring how much biomass they produce, by measuring the rates at which they perform these reactions. And so using these data of metabolite dynamics combined with some measurements of biomass, he was able to parameterize a simple model. So this model is just a consumer resource model that has 3 ODEs. The time dependence of biomass X, of nitrate A and nitrite I. And these have simple terms, right? So this is growth on nitrate in a mono form, growth on nitrite, consumption of nitrate, production of nitrite, and consumption of nitrite. And we can fit these models to these data, and that spits out for us a phenotype encoded by some set of parameters, RA, gamma A, RI, gamma I. RA is a rate of nitrate reduction. Gamma A is a biomass yield. How much biomass do I get per nitrate? And so on for nitrite. We can't fit these K parameters with the data that we have available, so we just fix them to a small value. So after a serious amount of work, aided by an undergrad in the lab, Derek Ping, Karna and Derek were able to acquire this massive data set of quantitative phenotypes. So these are vectors of the nitrate reduction rate, the biomass yield on nitrate, and so on, each measured in a whole slew of experiments for 80 different denitrifiers we isolated from soils. So what we want to do is take these quantitative traits, which are these rates and biomass yields, and somehow connect them to the genomic architecture of the system. And I would say that the sort of state of the art in doing this is maybe a flux balance model or something like this. It turns out these flux balance models are very hard to parameterize for many diverse wild strains. They work beautifully for model organisms, but for these organisms, they don't work well. And so we needed a simpler approach, again, to try to cut through this genomic diversity. There's a lot of genes in 80 genomes, right? Each one has, say, 5,000 genes. You're talking 40, 50, 60,000 genes. How do you tell what matters? What we're going to do is exploit the fact that we understand the mechanistic basis of denitrification. So this is this denitrification cascade I showed you before, and these are the enzymes that are responsible for these reactions. They're regulators and the transporters that are important for this process. What Karna did was he sequenced every one of these 80 isolates and then simply annotated these genes that I'm showing you here. There's about 17 distinct ones. And for each genome, which is a row of this matrix, he simply determined which gene was present or absent in each one of those genomes. So a circle means present, no circle means absent. And this gives us a 17-digit binary encoding of the denitrification genotype, if you will. Now, the reason this works for telling us something about traits, as I'll show you in a moment, is because bacterial genomes are highly streamlined. And the sort of old adage is use it or lose it. There's typically not a lot of extraneous junk laying around in bacterial genomes. They tend to be very streamlined. So you don't find often genes that are present and not performing some functional role. Okay, it's not 100% true, of course. There are definitely examples where that's not the case, but compared to eukaryotic or mammalian genomes, I think that's a fair statement. So given this, we made the intellectual leap to say, okay, let's take this genotype seriously and see if we can statistically map it to phenotype. And so what we did was we took this binary vector of zeros and ones, which told us which gene was present or absent in each genome. And we said, let's see if we can use a regression to map that genotype to these phenotypes. So these are the parameters of the consumer resource model as response variables. And we're going to do an additive model because we don't really have much more data than that. And beta ij is a regression coefficient. Gij is the presence of gene J in strain I, which has this phenotype Ri. And remember, this was measured in the lab previously as the rate at which this strain performs this reaction. And we have to do regularization and so on. I'm not going to go into the technical details here. But the bottom line is that this works remarkably well even out of sample. So what I'm plotting here is the predicted rate of nitrite reduction and the observed rate of nitrite reduction for about 50 strains in this library that perform this reaction. And the out of sample R squared is about over half. And so we have similar predictive power for other phenotypic parameters. So what this means is that if you give me another strain, and we in fact did this true out of sample test, if you give me another strain and tell me its genotype, and I go in the lab and measure how fast it does nitrite reduction, I'll predict it with, this R squared, just from the presence and absence of genes in the genome. I get asked a lot, does this work for other traits? And we're about to put up a new preprint in the next few days or so that examines that in more complex carbon catabolic phenotypes. The problem is harder there, but I think the approach still works. But why does this work? I mean, why does the presence and absence of genes tell you anything about the phenotype? Of course, the dynamics of this pathway are a complicated thing. How can we possibly, expect gene presence absence to be so informative? Well, it turns out, I think, that part of the reason is that the presence and absence of these genes has sort of conserved underlying mechanistic impacts on traits. So for example, there are two enzymes that do the second step in the cascade. One encodes the protein NERK and another one NERS. There's plenty of literature, many papers showing that NERK is much faster than NERS, both in vitro and in vivo as an enzyme. And what our regression recovers is that strains that have NERK do this reaction much more quickly on average. And so I think what's happening is, at least in part, not completely, this regression is sort of uncovering, in some sense, some coarse representation of some of the mechanisms that are present in the pathway. Certainly the interpretations are not so clean for all of these regression coefficients. Okay. So what I just did was I connected statistically genomic variation in the community to the traits of the organisms that are in that community. But I haven't really said anything about how these organisms sort of come together to perform this transformation of nitrate to nitric oxide. And so now what we want to do is we want to take this consumer resource model I already showed you and go there with it. So imagine I have the fitted metabolite dynamics for one strain and the fitted metabolite dynamics for another strain. Those arrows are characterized by this consumer resource model. And what we can simply do is in silico combine the action of these two strains using by numerically integrating these ODEs where two strains are present in a community from some initial condition. This gives us the zero free parameter prediction for what these metabolite dynamics should look like if two strains are present instead of one. Okay, assuming that the only interaction in the community is is governed by competition and cross-feeding of these electron acceptors. And the really remarkable thing in this paper is that these predictions work really incredibly well. So here I'm showing you the line is the prediction of this model with no free parameters, and the points are the data. Okay, so the fits are the predictions, not fits, are remarkably good. Here I'm showing you the quality of these fits for all combinations of 12 different species, 4 from each of these 3 phenotypes. And what you see by and large is that this matrix is white, and white means that this prediction error is low and that these predictions are quite good. There's one block here, interestingly, where that's not the case. And that block is in situations where we have one strain performing the first step of the reaction and another strain performing the second step of the reaction. And so for some reason, the splitting of the pathway across these two strains causes this consumer resource model to fail. And you can see this failure right here. So here's the prediction from the model, these lines, and the points are the data. So it's a pretty catastrophic failure. And we think we have some understanding. We think that there's a toxicity in this third species that's affecting the rate of nitrate reduction in the first species, and that throws off the whole prediction. So, but the punchline is, for the remainder of this matrix, all you really need to know are the traits of these individuals in a consumer resource sense, and you can make very quantitative predictions for what the metabolite dynamics look like in a community of, say, 2 species. So I don't want to spend a lot of time on it, but we generalize this to communities of up to about 5 strains, using a coarse graining approach to deal with these sort of non-additive interactions. And so we can sort of draw a box and treat these 2 weirdly interacting strains as a single entity within a larger community. And that coarse graining approach lets us get good predictions for communities of up to 5 strains. And so here the y-axis is this error in the prediction. And this is for cases where we've performed this coarse graining around these sort of non-additive pairs, if you will. Okay. So the punchline from this paper is that if we take a regression approach to connect gene presence and absence to the traits of denitrifiers, that those traits in terms of these consumer resource model parameters let us, for most interactions, make quantitative predictions of how these organisms are going to interact, and ultimately connects the genomes of those individuals to this flux of metabolites through the community, using these two, really three key ingredients I talked about at the beginning: a statistical approach to figure out which genes matter, quantitative measurements of traits, that is, these metabolite fluxes, and again, this simple consumer resource model to sort of close the loop. So this is a paper that was spearheaded by Karna Gowda, who's one of these postdocs that all of us PIs dream about, because he completely launched this huge section of my lab single-handedly, not single-handedly, with Derek, and an important collaboration with a good friend, Madhav Mani at Northwestern. Okay. All right, so that's one story where we connected genotypes to function. But now what I would like to do is to go back to this slide where I talked about why this problem of relating microbial community structure to function is hard. And one of the things that I said there was the environment is heterogeneous. And what I've showed you so far is examples in the paper with Alvaro and Mikhail Tikhonov, where we have synthetic communities in very homogeneous environments where we fix the environment in the lab and then we make some measurement of the function, and there it seems like we can make some progress. But in the wild, the environment is not such a simple beast, right? We don't get 96 well plates in incubators in the wild. And so how do we contend with this environmental heterogeneity? So really the question that I'm going to ask in this last part of the talk is, how do we map environmental heterogeneity to variation in structure? And the first thing to notice is that environmental variation drives patterns in structure. And this is something that we see in virtually every large-scale sequencing data set that's come out in the last 15 years. So here I'm plotting the abundance of a gene that encodes a particular enzyme as a function of soil pH. Doesn't matter what that enzyme is right now, but what you can see is there's a positive correlation. So for some reason, when the pH is more neutral, there's more of this enzyme. Okay, why is the question. Similarly, this incredible data set from the Tara Oceans project, where I'm just choosing one of the many patterns that they see. But what they see here is that the community richness has this sort of characteristic set of trends with temperature. Okay, these are just two salient examples that I chose, but there are also beautiful examples in human microbiome, for example, where you see patterns relating the composition of a community, say at the taxonomic, genomic, metagenomic, even transcriptomic level, and host phenotypes, disease states, and so on. So how do these patterns come about? I mean, what's giving rise to this pattern? I think our thinking about this problem really goes back to the foundations of ecology and to the work of Alexander von Humboldt, who wrote this very important book on the geography of plants in 1807. Alexander von Humboldt, we kind of take this for granted now, I think, because it's so deep in our collective sociology of science. But what he did was he drew a picture of this very famous volcano in Ecuador called Chimborazo. And what von Humboldt noticed was that as you go up the volcano, the plants change. Down here, there's big trees. Up here, there's tiny shrubs. And the argument at the time was that, well, the environment up here is hostile to big trees, and so the big trees can't live here, and the environment down here is not hostile to big trees. It's wetter and so on, and so the big trees live down here. And the idea there is that the environment filters out who can live where. And so basically the traits of the organisms in these ecosystems determine where they exist along this environmental gradient of altitude. So that idea now is what I would call environmental filtering. And remarkably, when you read these papers, such as the Tara Oceans project and so on, what you find is that environmental filtering is very often invoked as an explanation for some of these patterns, right? That at high temperature, we're seeing organisms that are high temperature adapted and so on. Recently, in the last 10 years or so, it's been asked whether this is actually true or not. It could be also that this is a collective effect, that there are some interactions in the communities that cause organisms to be able to thrive here and not here, and that the simple traits of each organism are not sufficient to understand why they exist in a particular environmental niche. This is probably best articulated in an opinion piece by Jonathan Levine's group at Princeton, where they ask, how much evidence is there for actually environmental filtering explaining these patterns? So we want to ask this question in microbial communities. And the strategy is as follows. I'll document a pattern relating structure to the environment in denitrification in soils. We'll use an enrichment experiment to recapitulate that pattern in the lab. That will let us dissect the processes that are going on in these communities. And from that, I will show that, at least in one case, the pattern that you observe is a collective effect, that the dominance of particular strains or species or genotypes in a particular environmental niche is arising from interactions and not from filtering. Okay, let's get started. All right, I'll try to finish in about 10 minutes. So we're going to use this data set from Per Bork's group, an incredible data set, almost 200 data points from these topsoil samples across the globe. And the really remarkable thing about this data set is that they measure all these environmental parameters, okay, so pH, nutrient levels, and so on. And they do shotgun metagenomics, so they can tell us what genes are there. We were given this data and we mapped those reads to these different genes that encode enzymes in the denitrification cascade. And what we get is, for each one of these samples, a bar which tells us this many reads map to this gene, this many reads map to this gene, and so on down the line, okay? And that gives us a data set that looks something like this. So here are the samples, here are the relative proportions of each one of these genes. And I can't go into the technical details for time today, but what Karna has done, and Kyle Crocker, actually another postdoc in the lab, is to use unit invariant singular value decomposition, which is a particular version of singular value decomposition that effectively normalizes the total abundance of denitrification reads in each sample, while preserving the correlations between the abundances of these genes in the sample. So what we're ultimately interested in is what proportion of reads are responsible for this first enzyme versus the second enzyme versus the third and fourth. What is the correlation between the enzymes that are being used in this denitrification cascade across the globe? So what this tells us is that, you know, in this sample, there's very little NAR, and in this sample, there's a lot of NAR, and that's correlated in some sense with which other enzymes are being used in each soil sample. Now, because we have environmental variables measured for each one of these soil samples, we can ask how the abundances of these genes relate to those environmental variables, again, using our friend regression. So we're going to ask, let's say for each soil sample, I have the pH, and I want to know if I can predict that pH by regression using the abundances of each one of these enzymes as an independent variable. And I'm going to do that for all the environmental variables that they measured in this data set. And what you find is that you can predict the pH from knowledge of the relative abundance of enzymes in this denitrification cascade with an R-squared of about 1/2, okay? So that means, at least in a correlation sense, pH is very important for determining which enzymes are used in the denitrification cascade in the wild. There's no causality here yet, but it tells us that pH is this strong structuring force, possibly. And this makes sense with what people know about soils. So I'm going to focus on a particular pattern that we observe in this regression, looking at the coefficients of this regression, and that is as follows. As the pH goes from acidic to neutral, the abundances of the gene encoding the enzyme NARG, goes down, and the abundances of the gene encoding the enzyme NAPA go up, okay? So there's sort of a trade-off. NARG is less well adapted at neutral pH, and NAPA the converse. So what we're going to do is do an experiment in the lab that recapitulates this basic pattern. So we take soil samples and we enrich them in denitrifying conditions at acidic conditions or at neutral conditions for multiple cycles in anaerobic conditions. And then we do sequencing and we look at who's there at the end. What we find is that in acidic conditions, Pseudomonas dominates, and neutral conditions, Rhizobia dominates. And sequencing shows us that this recapitulates the pattern I just showed you in the wild, because this Pseudomonas strain has this NAR enzyme and this Rhizobium strain has this NAP enzyme. And so this enrichment experiment is now recapitulating this global pattern, at least qualitatively. And we've repeated this on, you know, tens of different soil samples now. So it looks to be relatively robust. Okay, so at neutral conditions, we then take these two strains and grow them in acidic and neutral conditions. And what we find is that in neutral conditions, the situation is quite simple. The NAP genotype, this orange one, it dominates, just as we saw in the enrichment experiment and as we see in the topsoil data set. So this orange curve is telling me the relative abundance of NAP over multiple rounds of dilution cycles and the relative abundance of NAR. In acidic conditions, the situation is not nearly so simple. what we see is that this NAR genotype dominates, that's this Pseudomonas in blue, and this NAP genotype goes down, but ultimately stably coexists with this NAR genotype. And this is harder to understand in terms of an environmental filtering perspective. But it does square with what we see not only in the enrichment experiment, but also in the topsoil microbiome data set. So this NAR genotype is dominant. Is this filtering or is this a collective effect is the question that I'm going to ask. And what I'm going to show you is that filtering cannot explain this result. So now we grow each species alone in acidic conditions at pH 6. And what I'm plotting here is the biomass as a function of the cycles of growth. And what you see is that the genotype that dominates at pH 6, when we grow it by itself, it dies. Okay, so the biomass is going to 0, whereas the strain that does not dominate is doing fine. Okay, so this means that filtering cannot explain the dominance of this genotype in our enrichment experiment, at least. So what is going on? I will not drag you through all of the detailed experiments that Kyle Crocker did, but I will just show you what the mechanism of this is. So it turns out that this NAR genotype is a nitrate specialist. It does fast nitrate reduction, but that causes it to accumulate nitrite in the medium before reducing that nitrite. It turns out at pH 6, nitrite is toxic, and this NAR genotype is inhibited. This NAP genotype is the opposite. It's a nitrite specialist. It does slow nitrate reduction, but nitrite never accumulates. And that's why it's able to grow by itself in low pH conditions. So if we feed this strain, this NAR genotype, a bit less nitrate, it accumulates less nitrite, this red curve. And then when we compare how much biomass is produced in the high initial nutrient condition versus low initial nutrient condition, we see that it actually grows better when you give it less nitrate. And that's because it's accumulating less nitrite, and that toxicity of the nitrite is then alleviated, allowing it to grow to higher biomass. Okay, so what happens is that interactions in the community solve this toxicity of nitrite accumulation, and that allows this NAR genotype to dominate. So here's again this NAR genotype accumulating nitrite in the medium, essentially killing itself, causing itself to die in the monoculture at pH 6. But when we grow it together with this NAP genotype, what happens is the nitrate is still reduced quickly, but this accumulation of nitrite no longer happens. And the reason is that this NAP strain is sucking up the nitrite that the NAR strain is producing. So there's a division of labor that's happening in this community that's solving this toxicity problem. And that division of labor is allowing this NAR genotype to dominate. Okay. So that's sort of the punchline here. The punchline is that this collective effect, this interaction, allows this dominant genotype to exist at pH 6 where it couldn't by itself. And this is the pattern we see in the enrichment experiment and again at this global level. I want to save a little time for questions, so I won't go through this data, but we looked at these traits across a library of different strains that have these genotypes, and suffice it to say this specialization in the first or the second step, depending on which enzyme you use to perform this step, is something that we see ubiquitously across strains in acidic conditions. So we think that this specific example we studied with this pair of two strains that we isolated from this enrichment experiment is actually generic more broadly. I'm going to skip the data. So the argument here is that these global patterns in the abundance of these different denitrification enzymes as a function of pH is actually happening, at least in acidic conditions, through a collective effect, which emerges from interactions in the community. This paper is now in review. Okay, so I hope I've convinced you that this approach, this sort of three-tiered approach, can allow us to connect variation in the community in these complex levels of organization to the function, and ultimately really map complex to simple. So we're taking this approach now and looking at how to do it in very complex communities of soils, and I won't go into that now for want of time. Let me just briefly thank the people that are very important for the work that I showed you. The primary drivers are Karna Gowda and Kyle Crocker with some help from Milena. Luis and Kalmudi have moved on now from my group, but they did the closed ecosystem work. Derek Ping helped with the first project. And Kisuk Lee is the pioneer of soils in my lab now. These people were not mentored alone. I have longstanding and very fruitful collaborations with Madhav Mani, new work with Mikhail Tikhonov, Alvaro Arjun, Arvind and Vincenzo at UChicago and Otto at MIT. And these are the funders. So thank you so much for your attention. I'm happy to answer any questions.
Thank you, Seppe, for the nice talk. So before question, I stop recording.